Hello, and thank you for your interest in my poster. My name is Brian Royals, and I'm with the Department of Entomology and Plant Pathology at North Carolina State University. The title of my poster is Tobacco Thrips in Peanut Response to Imidacloprid and Fluopyram. I would like to thank my fellow colleagues for their help with this project. Rick Brandenburg, North Carolina State University, Sally Taylor and Sean Malone with the Tidewater Agricultural Research and Extension Center at Virginia Tech, David Jordan and Andrew Hare from the Department of Crop and Soil Sciences here at North Carolina State. It's a great team to work with. Suppression of tobacco thrips is important in North Carolina and Virginia to protect peanut yield. Tobacco thrips can also vector tomato spotted wilt virus in peanut. Systemic insecticides are often applied in the seed furrow at planting to minimize injury caused by tobacco thrips and the incidences of tomato spotted wilt. When suppression is inadequate to prevent significant feeding injury, acephate is broadcast within the first month after planting. Nematodes can also be an economically important pest on coarse-textured coastal plain soils where peanuts are typically grown. Aldicarb suppresses both tobacco thrips and nematodes and is considered highly toxic to humans and to the environment. Acephate, imidacloprid, and phorate are registered for thrip suppression only and do not control nematodes. A commercial product containing imidacloprid and a nematicide, fluopyram, has shown efficacy against both tobacco thrips and nematodes. In this poster, we will discuss results of research in these states comparing thrips injury and yield when imidacloprid plus fluopyram is applied in the seed furrow at planting compared when imidacloprid alone and phorate applied in this manner. We also determine the value in terms of thrips injury and yield of an acephate application broadcast 3 weeks after planting. So the location and the treatments. Experiments were conducted in North Carolina with 13 site year combinations with North Carolina and Virginia from 2014 through 2020. The cultivar Bailey was planted at the rate of 5 seed per foot and conventionally tilled raised seed beds at a depth of 2 to 3 inches in row. Previous cropping sequences included at least 3 years of corn or cotton planted prior to peanut. Production and pest management practices other than insecticide to control thrips were administered the same across all trials and were designed to optimize yield. Treatments consisted of a factorial arrangement of 4 levels of in-furrow systemic insecticides applied at planting and 2 levels of acephate applied to peanut foliage 3 weeks post-planting. Treatments applied the in-seed furrow at planting included no insecticide, imidacloprid alone at 0.31 pounds AI per acre, imidacloprid plus fluopyram at 0.31 pounds AI plus 0.21 pounds AI, respectively, and phorate applied at 0.5 pounds AI per acre. Treatment applied to the peanut foliage included no insecticide, acephate applied at 0.5 pounds AI per acre 3 weeks after planting, imidacloprid plus fluopyram were applied in 8-gallon water solution immediately after seed drop and before slit closure. Phorate was applied immediately prior to seed drop at 15 gallons of water solution using a compressed CO2 sprayer and used to apply acephate both in North Carolina and Virginia. Results from this study indicate that while imidacloprid plus fluopyram is less effective in protecting peanuts from thrips injury, protection was adequate to prevent yield loss. Also, while injury from tobacco thrips was reduced by acephate regardless of in-furrow insecticide, this reduction did not translate into yield increases. Collectively, these data suggest imidacloprid plus fluopyram can be applied to achieve adequate tobacco thrip suppression to protect peanut yield. While not addressed in this research, imidacloprid plus fluopyram can suppress soil parasitic nematodes and protect yield. Thus, nematode incidence is a factor when deciding if fluopyram may be beneficial. Although we did not sample nematode population, the rotations used in our trials likely resulted in lower and sporadic populations of soil parasitic nematodes. Similarly, tomato spotted wilt virus likely had a negligible impact on peanut yield in the study. I'd like to acknowledge the research was supported financially by the North Carolina Peanut Growers Association, the National Peanut Board, the Virginia Peanut Growers Association, the AMVAC Chemical Company. Appreciation is also expressed to staff at the Border Belt Tobacco Research Station, the Peanut Belt Research Station, and the Upper Coastal Plain Research Station, and also the Tidewater Agricultural Research and Extension Center for their assistance with our research. Thank you for your time.